and welcome back to our latest series, Pharmaceutical Excipients with Exceptional Purity and Performance, on the Croda Pharma YouTube channel. In our last episode, we explored our purpose, smart science to improve lives, our mission to empower biologics delivery, and the key role of sustainability in pharmaceutical manufacturing. Today we're shifting gears to focus on how Croda Pharma's super refined and HP excipient ranges enhance drug stability, and why it all starts with using the highest purity ingredients in your pharmaceutical formulation. So get ready as we take a deep dive into the critical role of excipient purity and why it's essential for your drug's stability, safety, and regulatory success. The excipients you choose to stabilize your active pharmaceutical ingredients are essential for unlocking bioavailability and achieving optimal drug absorption and metabolism. However, depending on the source of your excipients, they may come with impurities that can negatively impact the drug's stability, shelf life, efficacy, and even its safety to patients. Reactive species such as peroxides, aldehydes, and residual catalysts can accelerate API degradation by interacting with the drug substance, producing harmful byproducts that can compromise patient safety and therapeutic performance. These degradation pathways dramatically reduce the formulation stability and shelf life. Impurities are also heavily scrutinized during regulatory filings, with excipient quality being a critical parameter within the CMC portion of your NDA, BLA, or equivalent drug marketing application. Failure to meet stringent pharmacopoeial limits can complicate regulatory submissions by prompting requests for additional justification or data from regulatory authorities like the FDA and EMA. Selecting the right purity level for your particular pharmaceutical application is key. At Croda Pharma, we offer a full spectrum of excipient purity grades to meet the needs of both routine and highly sensitive applications. At the foundation, standard compendial grade excipients meet basic pharmacopoeial monograph requirements and are suitable for non-critical applications where high purity is not required. Taking purity one step further, our high purity or HP range is manufactured with tighter impurity controls and packaged under nitrogen to minimize oxidative degradation. HP excipients are a strong fit for oral and parenteral systems where enhanced stability is required. At the top of the purity pyramid is our super refined range. These excipients undergo Croda's proprietary purification process, adhering to the most rigorous quality standards in the industry. Our super-refined technology reduces oxidative impurities such as peroxides, aldehydes, and catalyst residues that contribute to API degradation. Super-refined products are also packaged under nitrogen to eliminate oxidative risk, offering the highest level of purity, stability, and batch-to-batch consistency, ideal for the most sensitive parenteral and high-risk biologic formulations. Together, these ranges provide formulation scientists with the flexibility to match excipient performance with the needs of their specific application. Croda Pharma's super refined and high purity excipients are trusted across a wide range of applications in both small molecule and biologic formulations. These include injectables, ophthalmic and otic drops, oral capsules, inhalation, nasal, and topical systems, to name a few. Our superfine range is often preferred for the most sensitive applications, such as parenteral administration, where purity is critical due to direct entry into the systemic circulation and bypassing of first-pass metabolism. Meanwhile, our HP-grade products are widely used in marketed injectables and oral formulations, offering enhanced stability across various dosage forms. And importantly, our excipients support the development of therapies not only for human pharmaceutical use, but also for veterinary health, where quality standards are increasingly aligned.
Hi, my name is Will Small, Research and Technology Specialist at Croda Pharma, and I'm excited to tell you more about our super refined excipients. Our super refined excipients are an extremely high purity range of pharmaceutical excipients produced using a proprietary purification process. This process reduces various excipient-related impurities to very low levels, including water, catalyst, and peroxides. The excipients are then stored under nitrogen, which helps to prevent oxidation and maintain the purity of the excipient over its shelf life. We even undertake additional testing beyond that required for monograph compliance, demonstrating the extremely high purity and low batch-to-batch variability of these excipients. This visual says it all. You can see the difference that super refining makes to the colour of these compendial excipients. The process significantly reduces the impurities responsible for colour, taste, and odour of the excipient. Super refining has a big impact on oxidative stability of excipients. Ethoxylated excipients, such as PEGs and polysorbates, can degrade in the presence of oxygen, firstly forming peroxides, which then go on to form aldehydes and low molecular weight organic acids. Here we can see this process for standard compendial PEG 400, shown as the solid lines on the chart. Peroxides and formaldehyde peak at around 8 weeks, before falling as they degrade into formic acid. Superrefined PEG 400, shown as the dashed lines, has much lower peroxide value initially, and because it is stored under nitrogen, then it doesn't degrade in the same way. Peroxides, formaldehyde, and formic acid are barely detected in the excipient, even after 24 weeks. Triglycerides, such as castor oil, can also undergo oxidation, with any unsaturated fatty acids particularly susceptible to degradation. The stability of standard compendial and superrefined castor oil was evaluated over 8 weeks at 50 degrees C. The peroxide value of the standard compendial oil was higher initially and then increased by 83% over 8 weeks. However, the superrefined castor oil, again stored under nitrogen and with any impurities reduced, is more stable with a much lower peroxide value both initially and at the end of the study. Preventing oxidation of excipients is crucial because the peroxides, aldehydes, and acids that they can transfer to the formulation can be detrimental to both API and formulation stability. Some excipients, such as polysorbates, can undergo hydrolysis, which is degradation caused by the presence of water. This causes cleavage of the ester bond, which both reduces the activity of the polysorbate and results in formation of free fatty acids, both of which are undesirable in applications such as monoclonal antibody formulations. Standard compendial excipients typically contain both water and some free fatty acid left over from the manufacturing process, shown here as the solid lines for polysorbate 80. Over time, the presence of water results in hydrolysis and causes the acid value to increase. However, the superrefining process removes water to almost negligible levels and also reduces the amount of free fatty acid, shown here as the dashed lines. Because there is very little water present in the superrefined polysorbate 80, hydrolysis is prevented, and so the acid value remains low. This makes superrefined polysorbates a great choice when formulating MABs. As well as being problematic for API and formulation stability, excipient impurities can also show up during analysis, so their removal in superrefined excipients can make analytical method development that little bit easier. For example, sesame oil can contain low levels of naturally occurring polyphenols, such as sesamin and azaronin, which are then virtually removed in our superrefined sesame oil. And synthetic excipients, such as isopropyl myristate, are often not chemically pure, and can contain residual starting material as well as other esters if the raw material is also not 100% pure. For isopropyl myristate, this means that the presence of some isopropyl alcohol, octanoate, and decanoate in the compendial grade, but superrefining removes up to 99% of these impurities in the superrefined Crodamol IPM. Plant-based oils, such as peanut and soybean oil, can contain proteins, which can trigger allergic reactions associated with those oils. In refined compendial excipients, those proteins are reduced to very low levels, but as the chart shows here, they are still present and measurable. But in the superrefined grades of these excipients, that proprietary purification process removes those low levels even further, below the limits of detection. So if you want the very highest purity excipient with the lowest chance of causing issues during your formulation development, then there are multiple reasons for considering superrefined excipients. Join us next time when we'll take a deeper dive into some of the benefits that superrefined excipients can bring to formulations, taking a look at stability enhancement of small molecules such as fulvestrant and docetaxel.